Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Abbvie, Apellis, Beacon Therapeutics, Janssen, MeiraGTx, Novartis and Roche. Thank you for watching. We hope you enjoyed this webinar and share your thoughts with us in the comment section below.
Now very excited to introduce our next speaker, who is Dr. Lesley Everett. So Dr. Lesley Everett has joined us all the way from the States. Very happy to have her here. She's originally from Port Angeles, Washington, and she completed her medical school and graduate degree training in human genetics at the University of Michigan Medical School. And she has, after completing her residency at the University of California and doing some other training, she joined the Casey Eye Institute as a faculty member in ophthalmic genetics and the retina divisions. So her clinical and basic research interests focus on inherited retinal diseases, genetic disorders of retinal vascular development, retinal vascular imaging, and novel gene therapy approaches for the treatment of degenerative diseases. Retinal diseases, sorry. So yeah, welcome, Dr. Everett.
Thank you. Can everybody hear okay? Okay, great. Well, first of all, it's wonderful to be here with you all this morning. And I wanted to start after this with no relevant financial disclosures to just say a special thank you to Fighting Blindness Canada and the entire team who organized the event today, and particularly for the invitation here to join you, and to Morgan, Jamie, Jackie, both Jackies, and to Larissa for inviting me to join you. So it's kind of a big topic to discuss stem cell based therapies for inherited retinal diseases and for age-related macular degeneration today in just a short half an hour. But I kind of want to provide a high-level overview for what is most relevant for patients and providers to know what has been done, where are we going as a field, and what are some of the most exciting developments that have been reported with regards to gene, cell, and molecular therapies for these degenerative progressive retinal diseases. So briefly, I'm going to talk about a little bit of introduction about IRDs, or inherited retinal diseases, and AMD, and all of your questions from earlier were just like a perfect segue to this talk. Then the current landscape of these different types of cell therapies, gene therapy, and optogenetics, and really who could benefit from which is sometimes very confusing for different patients. And then particularly focus on stem cell therapies for retinal pigment epithelial replacement, as well as photoreceptor replacement. And then round out and talk about induced pluripotent stem cells and retinal organoids as a model for clinical diseases and as a technology to test new therapeutics, including genetic therapeutics. If we have time, very briefly just discuss the difference between neuroprotection versus stem cell therapies and the role for those different approaches. And importantly, to discuss also some of the limitations and challenges for stem cell-based research. Of note, we just won't have time today, but I want to mention that there are also really exciting gene therapy approaches and work for retinal ganglion cell therapies for glaucoma that would be a whole nother talk in itself, and maybe we'll do that next year. So to start, there's pictures here on both sides for you, but I want to make sure we're all on the same page about what is the retina and the retinal pigment epithelium, because we're going to be talking a lot about these structures in the next half an hour. So as I describe to patients, the retina is like the film in the camera of your eye, and the retina lines the wall of your eye like wallpaper lines a room. And the retina is the tissue that's really responsible for taking all the signals and pictures that come into your eye and for turning them into a message that goes out the back of the eye through the optic nerve to the brain so you can see. And there's a clinical picture here on both sides of a normal retina with the optic nerve, the big yellow circle, the retinal blood vessels, and then particularly the macula, which is the area encircled by those blood vessels. Let me see if I can laser that here. The macula, which is our area of the retina that's responsible for our sharpest central vision. And of course, is affected by diseases like age-related macular degeneration and a number of other genetic retinal diseases. There's also a picture of the retina in cross-section on an OCT scan, which I suspect many of you have seen in your own clinical visits, which shows us the retina on its side, like we're looking at layers of the retina, like similar to taking a slice of a layer cake and looking at the layers of the cake on its side. And this is a picture of a normal retina through the macula, where you can see all of the different layers of the retina, the different cell types, and particularly out here in the back, these series of parallel bright white lines, which reflect where the photoreceptors live, the light-sensing cells, which are the rods and cones that are important for our vision and the function of the retina. And then the last bottom bright white layer here is the retinal pigment epithelial cells, which are a cell layer critical for the function and health of the photoreceptors and the retina. This is a schematic of a healthy retina in that cross-section in the cell types and layers on the left, as well as a diseased retina. This is an example for retinitis pigmentosa, but a similar picture might be true for age-related macular degeneration. And what you can see, and I'm going to show here in these pictures and describe to you, is that compared to a healthy retina in which we have intact photoreceptor layers, out here the rods and cones overlying the retinal pigment epithelial cells, in a retina that's had a disease or a degenerative process like retinitis pigmentosa or AMD, we see loss of the photoreceptors or unhealthy, not normal looking photoreceptors, as well as retinal pigment epithelial cells underneath. And this loss of these cell structures results in permanent and previously incurable in many conditions vision loss. Now, again, we're going to talk about cell and cell replacement therapies, particularly focusing on photoreceptors and RPE as they have to do with inherited retinal diseases and AMD today, but that's not to minimize the fact that, of course, many other conditions like pathologic myopia, toxicity, vascular events, and trauma can affect the retina as well. And it's important to note that all of these retinal structures, unfortunately, do not have the ability to self-renew or regenerate. And that's the big problem that we're trying to address with cell-based therapies, so that we can restore more normal structure and function to the retina when these cells are lost. Okay, so just as a little bit of background before we get into the research phase here and the clinical research that's been done, I wanted to describe to you what inherited retinal diseases are and age-related macular degeneration, which of course is familiar to many of you. So inherited retinal diseases, or IRDs, are caused by specific gene mutations or variants that affect how retinal cells function or how RPE cells function and survive in the retina. These are a leading cause of early onset vision loss in all populations throughout the world. There's a pretty big variable onset of signs and symptoms in terms of when these diseases present. But vision loss ultimately results from damage to the retina and the retinal pigment epithelial cells. Some IRDs are caused by just one gene or just a few genes, for example, choroideremia, just one gene, where other diseases like retinitis pigmentosa are caused by essentially up to 100 different genes that can be passed through families in different ways. We also know that many IRD genes just haven't been identified yet. We see a lot of patients in our clinic who clearly have a genetic form of an eye disease, but we just don't know their gene. And this is a schematic just demonstrating that defects in truly now hundreds of genes are known to cause incurable vision loss. And we categorize them clinically here, and you can see in this overlapping Venn diagram just the different types of clinical presentation or the different types of clinical retina changes we can see depending on which gene is involved. And so there's a lot of overlap too. One gene can cause different types of clinical pictures depending on the scenario and the type of mutation in that gene. Before we move on, I just wanted to show a few examples, some clinical pictures of the spectrum and the diversity of inherited retinal diseases we see in an ophthalmic genetics clinic. So in contrast to this picture of a normal healthy retina, we can see patients with very severe advanced inherited retinal diseases, such as Leber's congenital amaurosis, who have profound early vision loss. And there are shown here diffuse retinal changes that affect the photoreceptors and the retinal pigment epithelial cells. In contrast, there's a condition called Swordsby's macular dystrophy, also a genetic disease that results in significant atrophy and scarring and actually bleeding, similar in a way to age-related macular degeneration, but it's a genetic disease that is associated with one particular gene. And we actually treat this like AMD in the setting of needing injections. In contrast, a very different looking clinical picture here for best vitelliform macular dystrophy. We can see young children and older adults who develop these lesions under their retina, right in the center, called a vitelliform lesion. These can also result ultimately in atrophy and vision loss in the center. And then you might be familiar with Stargardt's macular dystrophy or ABCA4-related retinal dystrophy, which is one of the more common forms of inherited retinal disease that can have a big spectrum of clinical changes and severity in the patient population, including central vision loss. And then finally, a picture of the choroideremia that we discussed, which is an excellent condition that results in degeneration and progressive vision loss from the periphery to the center, affecting the choroid and the retina. Okay, now there's some parallels between some of these genetic diseases we discussed and age-related macular degeneration, which we've already discussed this morning a bit. But I wanted to draw on those parallels before we start talking about cell therapies for both of these type of conditions. So age-related macular degeneration is a leading cause of irreversible blindness and vision loss in Canada. Over 2 million, not 200 million people are affected. And it's more common, as you know, and as the name suggests, among older people, age-related macular degeneration, and Caucasian individuals. And of course, so therefore risk increases with age. There are some genetic risk factors that are known, but to the question that was asked earlier, it's different in these inherited retinal diseases where it's one gene and you have it or you don't. So it's a multifactorial condition. And the global cost of visual impairment due to AMD is about $343 billion. So a huge cost to the healthcare system and to patients. And RPE, or retinal pigment epithelial cell degeneration, is really a hallmark of this disease, as was already described and introduced. Many of you are familiar, as we also reviewed a little bit ago, with the two forms, non-exudative or dry AMD, which is the majority of cases, 80 to 90%. But until very, very recently, we haven't had any effective treatment for this. And the excellent question was asked over here just a bit ago about Syfovre, or pegcetacoplan, which is a complement factor inhibitor. And then that was just actually FDA approved in the United States this year and is now being rolled out in the clinic. And following shortly on its heels is another complement inhibitor of C5 that on the FDA side has had a new drug application accepted by the FDA. So for the first time, millions of people who are affected with dry AMD are going to be able to benefit from these therapies that have been shown to slow the progression of geographic atrophy and dry AMD. We also talked about exudative or neovascular AMD a little bit already. That accounts for about 10% of cases, but 90% of the blindness that's associated with AMD due to the bleeding, swelling, and scarring that can happen in the retina. Okay. And I think we've covered the background here of AMD and the type of changes earlier today, so I don't want to belabor the point, but in the setting of dry AMD, we can see RPE cell loss and loss of the overlying photoreceptor layers that causes vision loss, whereas in the wet form, we get abnormal blood vessel tangles that can cause bleeding and swelling in the retina and also result in scarring and vision loss. And this results in central vision loss for patients affected by this disease. Okay, so thinking about both of these diseases, I want to briefly discuss the sort of three main approaches that the field as a whole in ophthalmology and retina is working on to better treat and prevent vision loss for patients with both inherited retinal diseases and AMD. And this is such a hot topic and there's so much going on that in fact we have a whole day-long research summit just about this topic at the big ARVO meeting, which is the annual research meeting, which we just came back from in New Orleans about a week ago. And these include gene therapy, optogenetics, and cell therapies. I'm here today to talk about cell therapies, so we're going to focus mostly on that, but it really is important to understand how these all three play together and who could benefit from which of these therapies. And so, excuse me, for that reason, I've shown down at the bottom here a picture of the OCT scan of a healthy retina, in contrast, for example, to a retina with macular changes and atrophy secondary to ABCA4 mutations. And so the healthy retina, again, has all of the photoreceptor and RPE cell layers intact, whereas this patient with the ABCA4-related macular changes has an abnormal retinal structure loss of photoreceptors in the RPE, and a really, as you can appreciate, different anatomy of their macula. And so we have to think about which of these therapies or approaches would be most appropriate for each type of patient. Gene therapies won't work if there's not a cell to put a gene in to use it. So gene therapies are more appropriate for someone who has some cell loss but still has viable cells to be able to utilize a gene or a gene therapy. Optogenetics and cell therapies are going to be more beneficial for people who have more advanced structural changes, who have lost the normal cell structures and layers, for example. And optogenetics, as I'll briefly review with you, really entails sort of rewiring the retina and using cells that are not normally light-sensing cells to become light-sensing cells and provide vision at somewhat of a limited or more crude level than we would like, but better than their current vision. And then cell therapies, as we'll discuss in detail today and for the rest of my talk, are really looking at how we can restore more normal structure and function here by replacing either photoreceptors or retinal pigment epithelial cells. This is another diagram saying exactly the same thing, but emphasizing the point, and I'll go from top to bottom here, that really, again, the approach that we take is highly dependent on the status of the patient's retina and the cell structures that we have when we're meeting them in the clinic and what they could most benefit from. So people with fairly normal retinal structure and still present retinal cells or photoreceptors, but who are affected by a mutation in how those cells work, can benefit from gene therapy or gene replacement or gene editing. We can make those cells potentially work better. But as the retinal disease progresses, for example, and we develop fewer cells or more scarring or less normal structure, we're going to need to rely on those other therapeutic approaches like optogenetics, rewiring the retina, or cell replacement, putting in more normal cell layers or structure so we can help improve the quality of vision. So that's why I think it's important to understand how these things all work in complement and sequence to restore patients' vision over time. I'm going to just put 2 slides about gene therapy and 2 slides about optogenetics, but then we'll switch into cell therapy here. So this will be covered in more detail in the afternoon, so I don't want to cover everything that will be discussed later. But the premise of gene therapy is there's a genetic mutation causing a problem with the encoded protein in that gene that's critical for the function of the retina. So depending on the nature of that mutation, we have different ways to address that. If no protein is made because it's a frameshift mutation, for example, we can put a full new copy of a gene into a cell and it should be able to make that protein and improve the function of the cell. Whereas, for example, if there's a mutation that causes a protein to be abnormally folded or function abnormally, we can use gene editing or CRISPR, molecular scissors, to revise that sequence and make a more normal structure and function of that protein. Similarly, we can use RNA editing techniques to correct a mutation. Is that you're telling me I'm done? Okay. I have a long way to go. Of note, we can use both intravitreal injections and subretinal gene therapy approaches to apply these therapies to patients affected by inherited retinal diseases. We'll also discuss later, so I certainly don't want to steal the thunder, of course, about talking about successes of gene therapy. One is Luxturna for RPE65 biallelic mutations that cause profound early vision loss in young children, which has been very successful and is an FDA and EMA approved and is now utilized here in Canada. And then just recently, a patient who was treated at our center in Portland with another form of Leybert's congenital amaurosis, secondary to a gene mutation in the Gucci2D, for example, showed really surprising and excellent early results after her gene therapy. And she was able to see as a 17-year-old snowflakes for the very first time in her life. And her mother captured this video, which is available online if you'd like to watch it. So there's really promising work in the gene therapy space, which is complemented by the optogenetics approaches we also will just briefly review here. You can think of optogenetics approaches as using bioluminescent proteins, like in algae and jellyfish, and rewiring the retina to use those bioluminescent proteins to use alternate cells that are not normally the light-sensing cells of your retina to become light-sensitive. They can provide some level of vision, not perfect 20/20 vision, but restore a more normal ability for the retina to sense and respond to light. So there's an example here of the first group from Paris who has utilized this work, and they actually have restored some component of vision to a previously totally blind person who could just basically tell if the light was on or off. And utilizing this optogenetic system, Patients with this therapy are able to identify objects on a table or the crosswalk hashes and have a more improved quality of vision. We don't have time to discuss it here, but there's a number of different clinical trials for optogenetic therapies already in the works and a number coming along. Okay, so now to the main event. So stem cell-based therapies and what we can do with stem cells to restore vision and normal retina structure for patients with IRDs and for AMD. So stem cells are immature precursor cells with limitless self-renewal ability. These can differentiate into a number of different types of cells. And so they can make retina cells, they can make heart cells, liver cells, for example. We can think about two different types of cells, embryonic stem cells and induced pluripotent stem cells. Embryonic stem cells are just as they sound. They come from embryonic tissue, and we can actually isolate and harvest them from donor, for example, embryonic tissues or blastocysts, and use them to create new cell types. But there's obviously limitations with the availability and the ethical use of these tissues. So there's been great interest in reprogramming other cells that we can collect from our own bodies or of patients and turning them into stem cell-like cells that have that regenerative potential. And that is called induced pluripotent stem cells. In 2006, a group showed that we can take fibroblasts or skin cells or bone or blood cells or bone cells, and we can reprogram them into stem cells that we can then turn into photoreceptors or retinal pigment epithelial cells, which have tremendous possible implications for therapy and for regenerative potential in the retina. Again, these induced pluripotent stem cells avoid the ethical issues and the availability issues, of course, for the retina. And they're really ideal for patient-specific therapy because if we take, for example, my own cells, put them in a dish, correct a mutation, we can put them back in me without concern for rejection or immune-related complications. So for all of these reasons and more, stem cells represent a really phenomenal source for possible tissue regeneration for diseases affecting the retina, like AMD and inherited retinal diseases. And they're also really useful in the lab for studying human models of disease or studying new therapeutics or gene editing approaches so we can see how they work in the dish in the lab, but using real human cells that best reflect the human retinal condition we're trying to study, sometimes better than an animal model. So I walked through this picture here, but on the diagram on the right side is just an example of the two sources of these stem cells, so blastocysts and a true embryonic stem cell from human tissues versus, for example, a somatic cell, a fibroblast we took from a skin biopsy or for another tissue, and we can convert into an induced pluripotent cell in the lab. Then they can be grown and cultured and divided so we can have a limitless supply of these cells. And then they can be reprogrammed into any potentially tissue or cell type of interest that we would like to regenerate. So I'd like to start by talking about efforts that have been done to regenerate and replace the retinal pigment epithelial cells, that layer at the bottom of the retina that's really critical for supporting the health of the photoreceptors. Both efforts utilizing the human embryonic stem cells and induced pluripotent stem cells have been done with this work to develop RPE cultures and transplantable cells. With the goal here, as you can see, particularly from the diagram on the right, where we take a patient skin cell, for example, put it into the lab, grow it into a dish, reprogram it into what's called an inducible pluripotent stem cell again, and then send it down any number of pathways, but in our interest, retinal pigment epithelial cell pathway here, so we can grow retinal pigment epithelial cells in sheets, for example, on polymers, or as suspensions in the lab, and then introduce them back into the eye, either as subretinal injections or on a polymer sheet under the retina, or even through a different mechanism I won't talk about today called the suprachoroidal injection. Okay, so the very first, or one of the earliest examples of this approach was using human embryonic stem cells. So again, taken from humans, blastocysts and donor tissues rather than from patients and programming their cells to develop retinal pigment epithelial cells and replace them in patients with AMD and Stargardt's macular dystrophy. And they ran two prospective phase 1-2 studies to assess the safety and tolerability of this approach with subretinal transplantation of these embryonic stem cell derived RPE cells. They did 9 patients with Stargardt's macular atrophy who were 18 and older, and 9 patients with atrophic AMD who were 55 or older. They used 3 dose cohorts, 50, 100, and 150,000 cells for each of these disorders. And again, they grew these, sort of developed these as cell suspensions and then delivered them subretinally. I forgot to mention earlier, but please feel free to take pictures of all of this. And I've tried to write the critical information from these studies as well as have pictures just for your reference. But I know we all see and learn in different ways, so feel free to take photos of anything here. The main conclusions from this study were, first of all, and this was one of the earliest days that they did this, no evidence of adverse proliferation. They were kind of worried, are these going to turn into tumor cells or have unlimited growth potential or cause problems to the retina that we can't control. There was also no evidence of rejection of these cells when transplanted into the eye or any really serious ocular or safety events related to this transplanted tissue. But the adverse events in this particular study, which again used human embryonic stem cell-based cells, were more to do with the complications of the vitreoretinal surgery itself and then immunosuppression related. So dealing with the fact that these cells we put into the patients were not their own cells. In this study, 13 of 18 patients, or 72%, were found to have, and I'm going to show you in the pictures up here, some increasing subretinal pigmentation in the areas where these cells were put, which was thought to confer and indicate that these RPE cells were flourishing and viable and actually increasing in population or visibility throughout the retina in a number of pictures. In this study, actually, the best corrected vision improved in 10 eyes that received these transplants, remained stable in 7, and decreased in 1. So as the first attempt doing this type of work, this was fairly promising for safety and tolerability data. Then the next approach that built upon that initial study, the folks who are now at Wilmer as well as in California thought maybe we can do better by growing these human embryonic stem cell derived retinal pigment epithelial cells on a polymer. Rather than just putting a whole cell suspension under the retina, maybe we can grow them on a sheet to maintain a more sort of normal order and morphology. And then we can put that sheet under the retina to restore the RPE cell function and layer. So this group developed a composite subretinal implant to be used for people with advanced AMD. It's a polarized monolayer of these human embryonic stem cells that were generated into RPE cells on an ultra-thin synthetic parylene substrate here, which you can see down at the bottom right. Sort of looks like a bottle shape, but it's this polymer membrane which is designed to grow these RPE cells in a sort of more normal morphology and arrangement with a little handle that they can use to inject this under the retina. And this was tested in a single-arm, open-label, prospective phase 1/2 study with 16 subjects. And again, this sheet of grown cells was inserted subretinally in a retinal surgery. The interim first data that was published about the results for this were just 5 patients reported in 2018, 5 of the 18. And they found that on the OCT picture, the cross-section picture that looks at those layers of the retina, that it was consistent with the human embryonic stem cell RPE cells integrating well with the normal layers of the retina and the photoreceptors. None of the implanted eyes showed progression or vision loss. And one eye that was treated actually improved vision by about 17 letters, and 2 of the eyes that were treated were able to improve their fixation. Okay. There were no adverse events related to the implant or the surgical procedure. I'm going to move on a little bit from this. This study went on and they followed these eyes of the 16 patients actually over the next year and are continuing to study them. And there are, pressing the wrong button, clinical pictures here showing that all of these implants were successfully delivered under the retina. They stayed where they were supposed to be in the macula in the area of atrophy. And that actually, although it wasn't really powered to be the thing that they studied in this safety study, they were able to actually find that several patients, 4 patients, had improved vision through this study over the year follow-up. And one patient who passed away for unrelated reasons donated her eyes, and they looked at the tissues of these eyes after the implant and found that the implanted cells were still viable and present even 2 years after implantation without really any significant evidence of rejection. Okay, then the last thing to discuss, the allogenic RPE cells, or RPE cells from other people put into a patient, not their own cells, is I wanted to let you know that there's a current active recruiting study being done at the University of Michigan, which is a phase 1/2 clinical trial for the safety and tolerability of these embryonic stem cell derived RPE cells as a therapy for dry AMD. And these are actually stem cells programmed to be RPE cells, but they haven't totally fully matured when they're isolated. Following their isolation, they're differentiated into these mature RPE cells and transplanted into the retina, where the host RPE cells are diseased or have died. So if you'd like more information, the clinical trial number is there. Now, I'm going to move along quickly because I don't want to run out of time, but I want to mention briefly that work has also been done then to take a patient's own cells and convert them into retinal pigment epithelial cells and reintroduce that back into the retina as well. So therefore avoiding the issues of immunosuppression or rejection. The first attempt of this was in one patient in Japan with AMD, and they generated RPE cells from that patient's own fibroblasts. And these RPE sheets were actually found present and still doing well 4 years after implantation. The patient had stable vision from baseline and actually didn't require any more treatment or injections for the AMD. Subsequently, other groups have gone on to show that we can derive patient-derived induced pluripotent stem cell RPE cells from, for example, patients with retinal degenerations. We can induce them in the lab through a number of molecular signals to be these RPE cell cultures. They act like, they look like, they behave like RPE cells, and we could use those then for gene therapy to reintroduce back into patients later as a therapy. With regards, switching gears in the last few minutes here to photoreceptor cell replacement and regeneration, this has generally been more challenging for a number of reasons than RPE cell efforts. They have to both survive transplantation into the retina, but really then need to be able to make the connections and integration with the other cells of the retina to produce a visual signal. So there's a lot more structural and anatomic relationships that needs to be developed. Most of the work in the field of replacing photoreceptors has been done in animals rather than patients. And I just want to emphasize that we've now come to the point where we have the ability to derive from humans induce pluripotent stem cells, turn them into photoreceptors, grow them in anatomically normal photoreceptor layers and retinal structures, and then we have induced them and put them into a mouse retina, for example, where they've integrated into normal mouse retinal structures and developed appropriate cell-cell connections. As the next step, we've done similarly with rods, where they were derived from fibroblasts, using a combination of 5 molecules for reprogramming. They've put them into animal eyes and showed that the animals that were injected with these reprogrammed cells had more normal visual function by measuring their pupillary reflex in the dark and the light, and by measuring their tolerance, for example, to be in a dark space rather than a light space. Most rodents want to be in the dark, but not the ones who can't see well in the dark from their retinal disease. But when they fixed their RPE cells with this genetic therapy, the rodents actually who were treated preferred to return to the dark settings just as much as an unaffected or wild type mouse, showing that this restored some more normal retina function. And then finally, they've extended this work to show that we can grow retinal sheets and transplant them, for example, into non-human primates, which is a more physiologically relevant model to humans than mice are, and that we can grow these sheets into retinal layers and then isolate them after transplantation and show that they have integrated well into a non-human primate retina and made the appropriate cell-cell connections that are needed to function physiologically in the retina. Finally, the last step, really, before I think we can make really important gains in humans, is incredible work done by David Gamm and his colleagues, who David Gamm is funded by, Fighting Blind in Canada. And he's shown really through multiple different mechanisms here that we can demonstrate the synaptic connectivity of these induced photoreceptors in a dish, we can demonstrate that they have the ability to sense light and transmit this signal, which is an incredible feat after years of work of this whole field that's gone, as you have seen, from one patient who had an experimental therapy, for example, to be able to regularly, reproducibly generate and reprogram cells that we can ultimately put into the retina with light sensing abilities. As a cautionary note, just briefly though, of course, not all studies and early phase studies in humans and cell replacement succeed. And you might have heard of the Retina therapy trial not continuing. This was a retinal progenitor stem cell therapy injected under the retina, which unfortunately had a lot of surgical complications and was not continued due to those complications. I'm going to wrap up here, but I did want to just briefly mention retinal organoids as a very critical and useful model for understanding retinal diseases, for identifying novel therapies, and for ways to more rapidly test them in the lab rather than just waiting to see what we learn in human patients. So from a similar idea that we discussed, we can take fibroblasts or blood cells, we can reprogram them into the induced pluripotent stem cells, and we can help make them grow into mini retinas in a dish, which are called retinal organoids, which can then be used for all of those different purposes, therapeutic testing, gene editing, development, and for better understanding the pathophysiology or mechanisms of disease. And again, they more accurately reflect human diseases than, say, an animal model of some human conditions. We can scale these up and we can manipulate them easily in a dish and in the lab. And so we also can use these retinal organoids to understand the best way to optimize for gene therapy approach. For example, which vector to use to target which type of cell, which has the most efficient transduction efficiency. So when we do gene therapy, we can do it with the highest success rate for introducing new genes into the retina to fix the problem that they have. Finally, we just use them as models also to better understand the mechanism of disease. This is a retinal organoid model for one form of autosomal dominant retinitis pigmentosa resulting from mutations in PRPF31. They grew retinal organoids from control patients and these photoreceptors, and they could appreciate that the mutations in PRPF31 cause really abnormal cellular and morphologic defects of the photoreceptors. Then similarly, after learning the mechanism of why that happens, the people who did this study were able to treat by gene therapy, in addition, the organoid, this cell defect, and cause the cell morphology and function to be more normalized. So this is a very rapid way to test these therapies before we move to patients. I'm going to skip that one because it's a similar idea, but for another form of retinitis pigmentosa. And then finally, I think this is of relevance to Canada as well. We can use these retinal organoids to test the implications of genetic changes that are called variants of unknown significance. We get these results back from genetic testing, where we look for the mutations that cause an inherited retinal disease. Sometimes it's a slam dunk. It's a mutation that's known, and everybody who has that we know has RP or an inherited retinal disease. But sometimes we get back the test results that say variant of unknown significance, which just means we don't have enough data from patients yet to understand whether that's the mutation that caused the disease. With this type of approach, we can actually test that out in cells and see what was the consequence for either cell growth, normal function, or for example, splicing of the genetic material in the cell that tells the cell how to behave. And in Australia, where Luxturna therapy is also publicly funded, they've started to use this model to test and verify that these variants of unknown significance are real and are the disease-causing problem before they go spend $1 million for Luxturna therapy. And it's a really effective way to understand whose mutations are causing real disease. So in summary, just a few of the challenges and limitations. There's a lot of promising work, but of course a lot more to go. There are surgical procedure and complications to still be worked out to optimize these therapies. We have to really understand how to identify and manage immune responses, particularly when we're not using a patient's own cells for therapy. We need to better understand how to characterize the anatomical and functional efficacy of these therapies and even have a way to sort of tag or monitor where these cells go and what happens to them after they're put in the retina. And we need to have very long-term follow-up for safety and functioning of transplanted cells when we're using these technologies, as well as rigorous regulations and standardized protocols and ethical use of this technology, because unfortunately, There are some examples of stem cell clinics or places that patients can seek care, often outside of the United States or Canada, proposing stem cell therapy, for example, taking a patient's own adipose or fat tissue and reimplanting them into the eye as a therapy. This has been well reported now, unfortunately, to be unsafe and cause, in several cases, severe blindness and glaucoma problems that could have been avoided if the patients had known that that wasn't a regulated or well-studied known therapy. So it's important that we do all of this work, all this preclinical work and clinical trials to best understand how to do this safely and effectively. I think I hit on all of my conclusions here, and just for the sake of time, I'll wrap up and ask for questions, but I think this really shows that this is an incredibly promising field, and all of the work that all of the doctors have told you is coming, it's coming, it's coming, it is truly coming, and it's nearly ready for prime time in patients. So I'm delighted to share that with you, and I look forward to your questions. I'd also just like to thank our whole team at the OHSU Ophthalmic Genetics Center, my colleagues in our clinical and clinical trials divisions, as well as the funding for my research. With that, take any questions.
Thank you so much. That was such a wonderful overview. And I think some of the researchers and clinicians this afternoon will also touch and expand on some of these points. I'm sure there are questions out there, so I'm going to open up the floor for any questions anybody has. Okay, I'll start here close to me.
I'm curious, if you extract cells from my body to reprogram, how long does that lab work take?
That's a really great question. There's some different protocols here. The reprogramming itself, obviously the process doesn't happen overnight, and there's a lot of quality controls and parameters. That's usually about 40 days or so, as at least some of the protocols I showed, to go from, say, a skin cell to a bona fide acting like an RPE cell, looking like one. Where in contrast, to grow those whole retinal organoids, a whole retina in a dish, that actually takes months to get them, just like developing a human does. So it's a slow, sort of laborious process.
Thank you very much for a very informative talk.
Thank you.
In your slides, you mentioned several slides where you're comparing an RP infected retina and a normal retina. Would you be able to describe for people like me who are blind what those two images look like to an ophthalmologist who is looking at a normal retina? Like I'm imagining a nice pink uniform retina for normal, and maybe my retina is full of black spots or something. Can you just tell me in basic terms what those images look like? Absolutely,
and thank you for, that's a great question. So you're exactly right. It depends on which picture we're talking about, but for the color, ones we call color fundus photos, or how it looks when an ophthalmologist looks in your eye, a retina that's not affected by a retina condition actually does look quite pink, and we can see the optic nerve and healthy blood vessels, which are supplying blood to the retina. And then really in a retina that doesn't have one of these retinal conditions, most of the retina is quite homogeneous in color and appearance, and we can appreciate that sort of the health of all of the retinal layers are robust and intact. Whereas any of these conditions we talked about, like retinitis pigmentosa, inherited retinal disease, or AMD, can have a number of different clinical features, but pigmentary changes, areas of discrete atrophy or fibrosis we can see when we look in that are corresponding to those cross-sectional pictures we looked at, where some of the outer retinal layers or the retinal pigment epithelial layers are missing from the anatomical structure of the retina. And so different folks have different types of clinical presentations depending on what the underlying reason is, but you're right, they can have black spots, pigment changes, scarring, or areas that have had atrophy secondary to these changes.
Oh, no, I'll come here first. Say, am I walking back and forth?
Thank you. Hi, yeah, you mentioned something about fat cells you could use for the eye. I got lots. You want to use some, Mike?
I think we all want to do that. So in theory, actually, with the right molecular reprogramming, I think fat cells could be converted to different types of cells. But the thing I mentioned at the very end was a report, which have now been, unfortunately, numerous reports of patients seeking care at a clinical center that advertises itself as a stem cell therapy center. These are not part of academic or typical clinical ophthalmology centers you would encounter here, and they're not legitimate clinical trials, but they advertise to patients who understandably are desperate and looking for any and all therapy that they can perform stem cell therapy using that patient's own cells, which they isolated from their adipose tissue and injected into their eye. But that resulted in profound blindness and severe complications.
Hi, it's a very interesting subject and interesting paper, but because of my eyes, I couldn't see the slides and I couldn't read the comments. So I'm wondering if there's a publication that I can sort of look it up again and read it.
I think everything was recorded, and I'm also happy to share all of the slides, which I really built knowing that folks would like to probably revisit them today and read or look at them more magnified or at a slower pace. So I can certainly share those as well.
If I could have a copy of the presentation, that would be great.
Yeah, so Morgan will share the slides afterwards.
Okay, thank you.
You're welcome.
So yeah, Morgan can share the slides afterwards so everybody can spend more time if you want to use magnification.
Exactly.
And I think Dr. Everett also, there was a lot of information on there, but I think you sort of described the take-home messages quite well as well. But for people who want more information, we will provide it. I see a question over here. I'm going to walk over. Okay.
Hi, you were mentioning the fact that once you know what type of gene, I have RP, so once you know what type of gene you have, is there a cost to be part of these, I forgot the term, but for these clinics, and is there one here in Canada that, as Canadians would be able to sort of volunteer for?
Great, excellent questions. And I'll also ask Matt Benson to help me a little bit with that with regards to the Canadian resources. But I learned last night also just the differences between the United States and Canadian version of coverage for the genetic testing, which is also covered here. You usually have to have a referral to see your retinal specialist or an ophthalmic genetic specialist. But then there is coverage to have your genetic testing panel done so we can identify which gene, for example, is the reason for your RP versus someone else's. In the United States, we do a lot of that with sponsored testing that's sponsored through like My Retina Tracker, which is not of no cost to patients as well. And then the next step is really getting plugged in with an ophthalmic genetics specialist, for example, which are actually new developing clinics here. I've been learning as well in Canada, and more specialists who are really working specifically in this inherited retinal disease field. And then they, as well as just you as an advocate and learner online, are able to look at where are relevant clinical trials for my disease or my gene, all of which is at least nicely summarized certainly on the websites for Fighting Blindness Canada. I saw a nice comprehensive list of trials, as well as on the clinicaltrials.gov website, which has a comprehensive searchable list for clinical trials in the United States. And we actually have a lot of folks who come from Canada to Portland and are participating in our clinical trials as well. And to your last question, which I would also sort of emphasize is quite different than that stem cell clinic we just discussed, participation in a clinical trial, a legitimate randomized controlled clinical trial that's gone through all of these investigative steps never costs money to the patient or their participants. You shouldn't be paying money to enroll in those, and you shouldn't be felt like you're being pressured or recruited into them or doing something that you didn't want to do. So those are really important key distinctions.
Yeah, thank you, Doctor. That was a great answer. And if anybody has more questions about specific clinical trials or are there clinical trials for my eye condition, we do have a list of clinical trials for inherited retinal diseases. geographic atrophy, and some other innovative treatments on our website, but you can also contact our health information line, which Morgan leads, and she can help you navigate the clinicaltrials.gov website and identify if there are potential opportunities for you to participate, whether it's a treatment or even a natural history study, which is really important to learn more about the progression of a disease before some of these clinical trials are starting. Okay, I think that is it. I'm going to turn it back to Morgan now.